it is of the hour, so I'm going to go ahead and get started. So welcome everyone to a Zoomed Cancer Center Symposium. It is really my great honor to introduce Ashi Weeraratna. I had it down, Weeraratna, to give this talk. We've been trying to get Ashi out here for, I think, at least a year now. And everything that could possibly go wrong has gone wrong with that. But it's really fantastic. I've known Ashi for quite a while now because we're both in the cancer and aging realm. And first off, she's just an absolutely outstanding scientist, and you're going to see that from her work today. But first, let me give a little bit more of a formal introduction. So Ashi is currently the E.V. McCollum Chair of Biochemistry and Molecular Biology at the Johns Hopkins School of Public Health. She's also a Bloomberg Distinguished Professor of Cancer Biology and the co-program leader of the Cancer Invasion and Metastasis program at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins School of Medicine. So Ashi got her start actually in that same area of the country, getting her PhD from George Washington University Medical Center. She then went on and did postdoctoral research at where she is now at Johns Hopkins. And then she did a stint at the NIH and the NIA, which I imagine kind of got her interested into the aging realm. She then went on and was an assistant professor for, I think, 3 to 4 years at the Wistar Institute before being recruited back to Johns Hopkins into her current position as chair of the department. She's done just amazing work in the realm of understanding connections between the microenvironment and cancer progression. And I think her work in the aging area has been particularly interesting, showing how changes in collagen, lymphatic permeability as we get older can substantially influence the behavior of cancer cells with a real focus on melanoma in terms of how well they metastasize and so forth. And this is incredibly important because we know that depending on someone's age, they can have very different prognosis for the very same cancer. And so understanding these connections should lead to better interventions. And many of these interventions may even be in an age stratified manner. I'll end with something that I think is, and I partly get this because I follow Ashi on Twitter, which I encourage you all to do. But Ashi is such an advocate for others. And that by others, I mean like as a mentor and an advocate for students and postdocs and junior faculty and trainees. But also in the last number of months, I've really gotten to see how she's such an advocate for diversity. And we all know how important that is. And so she's not just committed to science, she's committed to making science a better place. So with that, I'm really happy to turn this over to Ashi and let her take it away. Thank you, Ashi.
Thank you so much, James. That's such a kind introduction. And I'm so sorry that I'm not there in person. I would have loved to be there, but I'm glad that we at least got to do this via Zoom. So hopefully there won't be like an electrical surge or something during the talk. So thank you again for having me out there. That's really nice.
Ashi, I forgot. Could I just mention one other thing? I forgot to say this, but please, everyone, if you want to ask a question, the best thing to do is just put into the chat. Just put, even if you want to ask it in person, just so we kind of have almost a list of names, so it's not a free for all at the end, just put, I have a question, or put your name or something like that. And at the end, we can just go through. But I always think it's nice for people to ask the questions in person instead of me having to read them all. Anyway, sorry, Ashi, take it away.
No worries. No, thank you. So, I hope that when COVID is over, you guys can come visit us in Baltimore, which is actually a much nicer place than some people would have you believe. It's very pretty here. And so, sorry, my computer's not cooperating. Okay, so as James mentioned, we've been very interested in aging, and we've been looking at this in the context of melanoma. And as many of you, I'm sure already know, melanoma is a very aggressive form of skin cancer. And it starts out on the surface of your skin, and by the time it invades to the dermis and into the subcutaneous tissue, it's aggressive and spreads to distal organs. So that's the number one problem. This lesion that starts out on the surface of your skin spreads everywhere. Not all patients are eligible for some of the great therapies we have available. And even those who are eligible, not all of them respond, and almost all of them ultimately relapse. So we're able to get sustained remission in patients in about 25% of patients currently. My lab's been really interested in understanding what drives melanoma metastasis, how these tumors resist therapy, and how the microenvironment plays a role. And that's really what I'm going to talk about today, specifically the role of the aging microenvironment. And I'm sure, as you know, by having James there and others, aging is really the #1 prognostic factor for cancer, poor prognostic factor for cancer. And you can see from these graphs here that if you look at cancer worldwide, the vast majority of individuals who develop cancer are over the age of 50, and certainly of those who die of cancer, the vast majority again are over the age of 50. And so as the population ages and, as we approach the decade 2050, over a quarter of people will be over the age of 50, this becomes a significant public health burden. So, melanoma is thought of as a disease or a cancer of younger people. Clearly from this graph, that's not entirely the case, although it's one of the most common cancers in younger people. You can see that as individuals age, the rates of melanoma increase dramatically. Furthermore, their ability to survive this disease, whether it's recurrence-free survival or overall survival, is decreased again according to age. So, this is obviously due to a number of different factors: changes in adaptive immunity, the chronic accumulation of genetic damage over time. But we've been very interested in trying to understand what's going on locally in the tumor microenvironment and how do all these normal populations of cells change as we age. We've been focused largely on fibroblasts. We're also looking at changes in different immune compartments. I'm not really going to talk about too much of that today, although I will touch on that. And the reason we're so interested in fibroblasts is because melanoma cells begin their life right here at this intersection between the epidermis and the dermis. And the dermis is really the part of the skin that is largely constructed of fibroblasts and the proteins that they release. So we wanted to know, and we also know that fibroblasts are susceptible to undergoing senescence. We know that there's positional stability in fibroblasts in the skin. So as we grow older, we don't have this constant turnover of fibroblasts. They tend to age with us. In the skin, at least, there's differences in other organs. And so we wanted to know how aging fibroblasts contribute to melanoma progression. So to do this, one of the first things we did was to create artificial skin. And we did that by taking fibroblasts from the upper inner arm of healthy donors. And the reason we chose that location is because that location is not an overly sun-exposed area. It's what we call an intermittently sun-exposed area. And we took fibroblasts from individuals between the ages of 25 to 35 and then 55 to 65. We use these fibroblasts to build this artificial skin. Essentially, it's this 18-day process. We learned this from our friend Meinhard Herlin at the Wistar Institute. And it's an 18-day process where you layer the fibroblasts, the collagen, keratinocytes, and melanocytes. So now we have this basal layer of skin into which we can put melanoma cells and ask the question, if we change the age of the fibroblasts and this reconstructs, what happens to the melanoma cells? And you can see quite clearly that melanoma cells remain tightly nested at the surface of these reconstructs in the young skin reconstructs, but in the older reconstructs, the melanoma cells start to invade into, what, if this was real skin, would be the dermis. We wanted to know if we could translate this to animal models. And so what we did was to use a mouse model of melanoma. This is the BRAF P16/P10 mutant mouse model created by Marcus Bosenberg and Martin McMahon. And what we did was to, these were back-crossed to C57 black 6 mice. The tumors were taken from these, dissociated, and turned into cell lines. And now we can take these cell lines and literally take half a flask and put half in a young mouse and half in an aged mouse. And that's exactly what we did. We then looked at growth, metastasis, angiogenesis, and several other parameters. What we found to our surprise was that in the skin of the young mice, the tumors actually grew faster. However, they metastasized to the lung much more effectively in the age mice. And so the question we had was, was there a difference in the growth because these tumors were invading so quickly from the skin of the aged mice to the lung, or were tumor cells moving at the same rate from the skin of the young and aged mice, but then growing out differentially in the aged? And so what we did to address this question, this is work done by a very talented postdoc in my lab, Mitchell Fain. And what he did was to inject tumors into the skin of young and aged mice. And so to define that, it's 8 weeks old for the young and anywhere between 12 to 18 months old. In this experiment, these were 18 months old for the aged. And so when Mitch goes back and counts the cells in the lung, so what he did was to label the cells so he could identify single cells within the lung. He sees that at 3 weeks, there's just as many cells in the young lung as there are in the aged lung. And these are usually in little single cells implanted all over the lung. However, if he waits just a few more weeks and he looks at these mice again, now he finds that in the young mice, these tumor cells are still sitting there as single cells, but as you can see in the aged mice, they've started to grow out dramatically. And so Mitch was able to do that over a series of mice. And so what he wanted to know was it possible that the aged lung fibroblasts were impacting the proliferation rate of these melanoma cells? And so he sort of modified our skin reconstruct to make what we call a sandwich reconstruct. And he layered in fibroblasts, collagen, and he stuck the melanoma cells in the middle. These were GFP tagged. And then he just gave them some time and he compared the growth rates of the melanoma cells between the young and the aged lung fibroblasts. And I think you can see quite clearly that melanoma cells grown with aged lung fibroblasts proliferate much more rapidly. And this is quite the opposite to what we see in the skin, where they're growing again a lot faster in the young mice and they proliferate more rapidly in the presence of skin fibroblasts. So for us, this was really our first clue that, fibroblasts in different organs could have different impacts on tumor cells. And Mitch wanted to explore this a little further, try to understand what was driving this observation. And so he took our secretome analysis of this young and aged skin fibroblasts and compared it to a new secretome analysis of young and aged lung fibroblasts. And what he found was there was a striking difference in one particular family of molecules, our old friends, the Wnt signaling molecules. And he found that in the aged skin fibroblasts, there was more of a non-canonical Wnt signaling phenotype as typified by things like Wnt5a, SFRP2, DKK1, and RR2. But in the aged lung fibroblasts, that sort of switched over to a more canonical Wnt signaling phenotype with molecules such as SFRP1. And so we were quite struck by the fact that SFRP2 showed up in the aged skin, but SFRP1 showed up in the aged lung. And so we wanted to know what that meant for the melanoma cells. We had previously shown that when aged fibroblasts secrete SFRP2, they drive Wnt signaling, they inhibit canonical Wnt signaling, reduce levels of beta-catenin, and can also increase Wnt5a expression. On the other hand, when we now went back and we treated melanoma cells with recombinant SFRP2, they decreased levels of Wnt5a, increased proliferation. And when we took these experiments into the mouse, we found that if we treated aged mice with an antibody against SFRP1, we could inhibit the outgrowth of melanoma cells in the lungs of these mice. So this kind of went hand in glove with something we've known to be a phenomenon, if you will, in melanoma, which is that you require both arms of the Wnt signaling pathway. So you have the canonical Wnt signaling pathway, which is critical for the bypassing of melanocyte senescence, for driving growth and proliferation. And then as cells become more invasive, you see an increase in non-canonical Wnt signaling, specifically Wnt5a, and that drives a highly invasive but very slow cycling phenotype. These cells are also therapy resistant, in part due to the fact that they're not cycling very rapidly. The other interesting thing about Wnt5a high cells is that they carry markers of dormancy. And so when we first started looking at the relationship of Wnt5a to dormancy, we turned to our good friend Julio Aguirre-Guiso, who's at the Mount Sinai School of Medicine, and got from him basically a signature of markers of tumor dormancy, and then compared that to levels of Wnt5a. What we saw quite consistently was using Julio's signature. We could see that almost every gene that he's found to be associated with dormancy was highly expressed in Wnt5a high cells, but not Wnt5a low cells. When we look at markers of proliferation for melanoma, including the MITF gene and MART1, we find those to be highly expressed in Wnt5a low cells, but not in Wnt5a high cells. And this is TCGA data, so these are patient tumors. So we wanted to know if there were age-related changes in the lung that drove the signature from from a dormancy versus proliferative phenotype. And what we saw was that when we take melanoma cells and we expose them to conditioned media from young or aged lung fibroblasts, we see that there is a very distinct downregulation of markers associated with dormancy, such as AXL, NR2F1, et cetera. And if we look at markers of proliferation, those come up, which is, very consistent to what we're seeing both in our in vitro and in vivo growth studies. And so this tells us that the age microenvironment in the lung is taking our melanoma cells and driving them from a sort of slow cycling, almost dormant phenotype in the young lung to a rapidly proliferating phenotype in the aged lung. To sort of pin down the role of Wnt5a in this, because, until this point, we've always said, oh, Wnt5a is so important in metastasis. And here now we were finding that actually in the lungs, we needed to turn off Wnt5a for outgrowth. And so it turns out that this is so much more plastic than we initially thought. And what we did was to make an inducible cell line that we could turn Wnt5a expression, this is the upper band, off using doxycycline. And if we turn dox off very early, we actually see that there is a decrease in the initial dissemination of the cells from the primary side of the skin to the lungs. But if we turn dox off 3 weeks later, after the cells have had time to get to the lung and then started to grow, we can now increase, this is in young mice. So we can now increase the outgrowth of tumor cells by turning off Wnt5a. So just again, to summarize that, because I know it can get a little confusing, if you have a tumor that's growing in the skin in a situation where it has where cells have high Wnt5a, they'll migrate to the young or the aged lungs. And in the young lung, they tend to not grow out because they continue to express the Wnt5a. But in the aged lung, the SFRP1 is secreted by the aged lung fibroblasts. This shuts off the Wnt5a signaling pathway, increases the ability of these cells to grow out. So, basically our current working hypothesis of this is what I just said. In addition, we are looking at changes in the immune microenvironment in the aged lung. And what we're seeing is that there is an increase in myeloid-derived suppressor cells, as well as Tregs in the aged lungs in the lungs of aged tumor-bearing mice. So I'm sorry, I'm being distracted by my dog. Go. So one of the joys of working from home. So So this is just one of the changes that we've seen that aging can affect. And we've also seen that aging can affect multiple other aspects of tumor progression, including matrix changes, changes in response to therapy, angiogenesis, changes in metabolism. And I'm going to touch on a few of these as I go through. So one thing we had noticed, another role for SFRP2 that we had noticed, I apologize for the background noise, Amy, come on, was that tumors in the skin of aged mice have far more angiogenesis than tumors in the skin of young mice. And when we look at whether that is new angiogenesis in the age mice or the young mice, we find that there's a lot more active ongoing neoangiogenesis in the tumors in age mice. If we take endothelial cells, these are dermal microvascular endothelial cells, so they're the endothelial cells you find in the skin, and we incubate them in fibroblast media from young fibroblasts or age fibroblasts, we find that the HMVEC cells in the age conditioned media will make tubules much more effectively. This is just a quantitation of that. However, when we look at VEGF and its receptors in patients over the age of 65, we find that there is a decrease in VEGF expression and its receptors in older patients. So we're seeing an increase in angiogenesis and a decrease in VEGF, and that didn't make any sense at first until we remembered that SFRP2 has been shown by this group down in South Carolina to be a potent stimulator of angiogenesis. And so we wondered, since we were seeing SFRP secreted in the age microenvironment, and we knew that it could impact metastatic progression, we wanted to know whether SFRP2 might be sort of taking over as a factor for VEGF in our tumors in the age microenvironment. So to answer this question, Mitch paired up with Brett Ecker and Aman Prikar in the lab, and they started to look at the impact of SFRP2 on the growth of blood vessels. So they again took their HMVECs, they treated them with conditioned media from younger age fibroblasts. Again, they saw the same thing. And now if they manipulate SFRP2, so they add SFRP2 to the young, or they deplete it using an antibody from the age, they can completely impact the growth of these blood vessels. They can do this in vivo as well. And so these are young mice that are treated with recombinant SFRP2. They dramatically increase their angiogenesis. Old mice have more angiogenesis, and now if you treat them with an antibody against SFRP2, they dramatically decrease their angiogenesis in the tumors. If we look at VEGF in correlation to SFRP2, you can see what the patient data told us, that in the tumors in the young mice, there is more VEGF, less SFRP2, and in the age mice, that's reversed. So this really made us, take pause and say, well, what does that mean for treatment and some of the therapies that we have? In melanoma, Avastin is used widely. It's an anti-VEGF antibody. And so that suggests that, that may not work in older patients since they start to lose expression of VEGF. So it turned out that some colleagues in the UK had run this very large clinical trial of over 1,300 patients, where they looked at response to bevacizumab, which is the antibody against VEGF. And they concluded at the end of their study that there was no change in overall survival. And so what we did was to contact them and ask them if they would mind reanalyzing their data, and this time to really parse it out by different age groups. When they did that, what they found was that actually patients under the age of 45 did much better on Avastin, and patients, as they predicted, 65 and older did not do that well. So, so these patients had a 30% chance almost of doing better on Avastin. So our theory then was that, these patients are doing better on Avastin because they actually have VEGF, so you can actually target it. And that implied then that if, as they get older and they have SFRP2 and less VEGF, obviously Avastin is going to be less effective. We wanted to mimic that in the mouse. And so we took our young mice, we gave them an antibody against VEGF, which reduced angiogenesis as expected. And we also gave them recombinant SFRP2, which increased angiogenesis as expected. But now if we treat with anti-VEGF in the presence of SFRP2, we can't overcome that, and these tumors have a lot more angiogenesis. So just to summarize that, SFRP2 sort of takes over as a predominant angiogenic factor in the age microenvironment, and promotes angiogenesis during aging, and can really inhibit the efficacy of the anti-VEGF antibody in vivo. And the clinical trial analyses suggest that actually younger patients do have response to Avastin, and so it might be worth revisiting that clinical data even further. We're also really interested in understanding what's going on in the matrix around these blood vessels. Is it just that there are more blood vessels, or are they also less stable, more permeable? And so that's something that we're investigating now. And I'm gonna switch gears and talk about the matrix for a second, and then I'll come back and tell you about our investigations of the matrix on the blood vessels. So why are we so interested in the extracellular matrix? Well, it's one of the most visible effects of aging. US presidents are a great model for premature aging. You can see that the two things they develop are gray hair and wrinkles. And the reason that they develop wrinkles is that collagen and elastin are very tightly cross-linked together. They're bound together by molecules of high hyaluronic acid. But as we age, these cross-links start to break down. We lose levels of high hyaluronic acid. And that's when the skin sort of folds in on itself and becomes what we see as wrinkles. So Aman Kaur in my lab wanted to investigate this further. And so she took her skin reconstructs, which again, as you can see, the tumor cells in the skin reconstructs are more invasive, and she looked at the collagen density. She could see that using 2-photon microscopy, the collagen was much denser in these young skin reconstructs than the aged. But of course, since there was so much more tumor invasion, we wanted to make sure that it wasn't just the fact that the tumor cells were eating up the collagen. And so she took just normal mouse skin from young and aged mice and looked at the collagen there. And what she found was that in the aged mouse, the collagen was really differentially organized than it was in the young. We wanted to understand why this was, and so she went to the secretome data again, and she found that the number one protein that is differentially expressed between the young and aged skin fibroblasts is this protein called haplin-1. It is a molecule that you can think of essentially as the knitting needles that knits high hyaluronic acid together with proteoglycans. And so Aman wanted to know what the role of haplin-1 might be in knotting up the collagen in the skin. So we looked at, again, the skin in young and aged mice. And if she takes haplin, and if she treats these aged mouse skin with haplin-1, she can now start to increase the density of the collagen, very similar to what we saw with the aged mice. We wanted to understand even more about the ECM beyond just the collagen. We wanted to know how some of these ECM fibers might be oriented. And so we turned to my good friend Etika Kierman, who's at Fox Chase, and we asked her to help us with that. So Etty has these beautiful assays that she's designed where she allows fibroblasts to lay down a matrix, and then she goes in and she images that matrix from different angles, and the light refracts at different angles, and then each of those refractions essentially is assigned a color. So the more crisscross the fibers are, the more colors you see. The more aligned the fibers are, the fewer colors you see. And so you can see that in young fibroblasts, you have this very sort of crisscross organization of fibers with them going in multiple directions. It looks like a rainbow. But if we deplete haplin-1 from the fibroblasts, they now start to organize their matrix in a very aligned manner. If we look at matrix from aged fibroblasts, we see that they start out in this very aligned manner. And if we replace haplin-1, they then start to become a lot more colorful, i.e. they're organizing their fibers in a much more crisscross, less aligned manner. If you manipulate haplin-1 by depleting it in the young, you cause an alignment. We can reconstitute that by treating with recombinant haplin-1. And if we use denatured haplin-1 in the experiments with the aged fibroblasts, we don't see an effect. So it tells us that this alignment of the fibers can really be affected by haplin-1. So we were interested also in a parallel study. Brett Ecker, who was a visiting clinician to the lab for a couple of years and was incredibly productive while he was there, He looked at how these matrix changes in the ECM could affect the actual integrity of vessels, and he was looking at lymphatic vasculature. So his hypothesis was that when he looks at the ECM in the young and the ECM around the lymphatic vasculature, the integrity of that ECM is critical for maintaining endothelial cell-cell interactions. And he hypothesized that as we aged, the ECM would break down, these endothelial connections would become unstable, leading to an increased escape of melanoma cells from the lymphatic vasculature and the lymph node itself. And this is very much in keeping with clinical data showing that actually older melanoma patients have far fewer lymph node metastases than young patients, but far more distal metastases than young patients as well. So building on this, Gloria Marino in my lab wants to see what that looks like for endothelial cells. So she's just beginning to look at this. She can look at blood vessels in these young in age animal tissues and see that, they're sitting in very different looking collagen matrices, because of course the collagen is quite different between these young and age mice. She can see that if she gives them recombinant haplin 1, she can change the density of the collagen around them. I love this picture because it shows that, here's a blood vessel in an age mouse. You can sort of see, and I know this is completely unscientific, but I'll show you other assays that are scientific. But the reason I love this picture is because it looks like the blood cells are all knotted up in the vessels that have haplin 1 and kind of floating around in the vessels that don't. Obviously, that's probably just a sectioning thing, but it was really kind of cool to see that. So in order to examine this more closely, what Gloria did was to take young fibroblasts and ask them to lay down their matrix. And then she plated endothelial cells on top of that. The hypothesis being that endothelial cells on a young matrix would have nice V-cadherin connections because they were also very stably anchored to the matrix that those fibroblasts had laid down. Conversely, if she takes her age fibroblast, she hypothesized that, the matrix that they lay down is less amenable to these nice tight V-cadherin connections. And so she looked at endothelial cells laid on top of these matrices, and indeed, that's exactly what she found. The same endothelial cells laid down on matrices from young fibroblasts had these beautifully V-cadherin zippers between the cells that was lost during aging. Gloria hypothesized that restoring haplin 1 could affect that. And so, she looked at, she took young fibroblasts, knocked down haplin 1, asked them to lay down their matrices, then plated endothelial cells on top. When she depletes haplin 1, she loses these V-cadherin connections. If she does the opposite, replaces haplin 1 in the age fibroblasts, she now sees these beautiful V-cadherins restored in the endothelial cells. So she went back to her mouse studies and wanted to know whether haplin 1 could actually affect the initial generation of the angiogenesis as well. And so she took her aged tumor samples, she treated the mice with haplin 1 and looked at the blood vessel formation. She's looking at CD105. So again, she's looking for neoangiogenesis and finds that actually neoangiogenesis is reduced by using recombinant haplin 1. If she looks at V-cadherin, she sees that also the stability of the vessels are decreased because the V-cadherin expression is decreased in the age compared to the young, and again can be restored by haplin 1. Finally, she wanted to look at what that meant for barrier integrity between the endothelial cells. And so in collaboration with the Isinger-Matheson lab at Penn, Gloria used an electrical impedance assay, which is basically an electrode array on which she plates her fibroblast-derived ECM. She plates endothelial cells on top of that, and then she measures current that can flow. The more resistance there is, the tighter those endothelial cell to endothelial cell barriers are. So in her control, where she plates the endothelial cells on gelatin, you can see that there's a ton of resistance, meaning you have these really tight barriers. Cells plated, the same cells plated on a young CDM also have good impedance and tight barriers, but the cells plated on aged ECM do not have coherent cell-cell connections. However, she takes the CDM, treats the fibroblast with haplin-1, and then places the endothelial cells on top of that fibroblast. She can restore the impedance and increase that barrier integrity between the endothelial cells. So Gloria's hypothesis right now is that haplin-1 is affecting multiple aspects of tumor cell invasion, from the primary tumor, decreasing the intravasation of those cells, affecting in the age the permeability of both the lymphatic and the endothelial vessels, so that even though you have more blood vessels, they're leakier, and so tumor cells can get in and out and metastasize to distal sites much more easily. And we're hoping that we're going to start to look now at the connection between haplin-1 and SFRB2, because we have some indications that there might be some regulation at the proteoglycan level going on there as well. Sorry, I'm beginning to lose my voice. And again, in the aged lung, just to remind you that, this phenotype switches, and SFRP1 can drive an outgrowth of these melanoma cells. So I'm going to switch gears one more time and tell you a little bit about Gretchen's work. So Gretchen finished her PhD in my lab and then insisted that she wanted to stay on for her postdoc against my advice, but she's very persuasive. So, and I'm delighted to have her because she's amazing. So in Gretchen's PhD work and work that she's continuing through to her postdoc, she's been really interested in trying to understand how the metabolism of fibroblasts can affect the metabolism of tumor cells. One of the things that she noticed early on was that older fibroblasts uptake a lot more glucose than younger fibroblasts, and she wanted to know what they were doing with it. So she looked at a number of different things, but the most striking finding was that aged fibroblasts have more Bodipi staining than young fibroblasts. This is in low glucose conditions. In high glucose conditions, it's even more dramatic, and those lipids are all over the place. But if she first blocks this glucose transporter and then looks at the lipids, even in high glucose conditions, she can really reduce the amount of lipids in these cells. What she wanted to know was what those fibroblasts were doing with those lipids and how those could affect melanoma cells. So she took her aged fibroblasts, she took conditioned media from them, and she put them on melanoma cells. And she found that consistently, if she treats melanoma cells with conditioned media from aged fibroblasts, they increase their levels of Bodipi, implying that they have more lipids in them. She did an extensive lipidomics analysis of the fibroblasts as well as the melanoma cells once they've been exposed to different fibroblasts, and found that, first of all, there were more lipids being secreted by aged fibroblasts, and they were the same ones being taken up by melanoma cells. So Gretchen wanted to confirm that in a few different ways. So she did a few different experiments. This is one of my favorites, where she stably labeled Bodipi, and stained her fibroblasts with them, washed them really well, and then co-incubated them with melanoma cells with a GFP tag. So in the images I'm about to show you, the green cells can only be melanoma cells. Red staining can come from are likely fibroblasts and can come from fibroblasts. And what Gretchen sees is when she incubates her melanoma cells with young fibroblasts, they don't take up too much of the lipids. They are taking up some for sure. However, when she incubates them with the aged fibroblasts, that increases dramatically. And you can see that there are loads of these cells with red cytoplasm and green nuclei. And almost every single cell has some level of red staining, telling us really that these cells are taking up these lipids, which was confirmed by the lipidomics as well. So Gretchen wanted to know how these cells were taking up the lipids, and she looked at a bunch of the different fatty acid transporters, including the classical CD36, as well as this family of FAT-P reporters. She found that almost all of them, except for FAT-P2, and I'll come back to that in a second, were unchanged in samples in in patient tumors with age. And so Gretchen, the only one that wasn't unchanged was FAP-P2. And she found that her melanoma cells, when exposed to media from aged fibroblasts, had increased FAP-P2. In the reconstructs, they had increased FAP-P2. In tumors in the mice, they had increased FAP-P2. And in patient samples from TCGA, FAP-P2 was upregulated with aging. And so this implied to Gretchen that, FAP-P2 was really the transporter responsible for the uptake of these lipids from the age microenvironment. So what she wanted to know was what impact that might have on the melanoma cells, as well as, things like their response to therapy and metastasis. And so she depleted FAP-P2 from the cells, and then she showed that if she took conditioned media from aged fibroblasts, they were no longer as effective as taking up lipids. She can also do this with a drug called lipofumata. And again, if she pretreats her cells with lipofumata, then exposes them to age-conditioned media, the melanoma cells are no longer as effective as taking up lipids. One thing she noticed when she was doing her FAP-P2 expression studies was the fact that melanoma patients who receive a targeted therapy that is commonly used for melanoma, so this is a therapy against the BRAF oncogene, she found that patients who did not respond well to this therapy and who quickly relapsed had increased levels of FAP-P2. And so she wanted to know, was it possible that by taking up these lipids, melanoma cells could resist therapy? So this was something that we have seen in the past. We have seen that patients over the age of 65 are less likely to mount a complete response to therapy against this drug. And if we look at some early animal experiments that we did, if you treat with an inhibitor against the BRAF oncogene, in young mice, the tumors regress, but in aged mice, they don't really respond. Now, I have to say that when we first did these experiments many years ago, this was a really striking result to us because you had one mutant oncogene and one drug targeting that oncogene. And we figured nothing else should really matter, but really even just, the age of the mice, the age microenvironment really affects the ability of those cells to respond to that drug. Remember, these are genetically identical cells being put at the same time in a young or an aged mouse. So Gretchen wanted to know what would happen if she manipulated FAP-P2 and stopped the uptake of these lipids. So she created a cell line in which she could inducibly knock out FAP-P2. And then she implanted these in a young mouse and treated them with inhibitors against the BRAF oncogene in the presence and absence of this gene. So the first thing she saw is that knocking down this gene doesn't really affect growth one way or the other in the young mice. In the young mice, when we treat with the newest generation of these BRAF MEK inhibitors, we see that the tumors will regress and eventually they come back. Excuse me. Now, if we do the same thing in the presence, I'm so sorry, 1 second.
Ashi, normally at this point, someone would be rushing up with a bottle of water, but.
I have the worst fall allergies. Sorry. Okay. The most dramatic experiment. It always happens. So this is what 10 hours a day of Zoom does. So what we found, what Gretchen found was that when she depletes FAP-P2 in these experiments, there's absolutely no change. However, when we look at the aged mice, it's completely different. We see that, the inhibitors have very little effect on tumors in the aged mice. They slow down for a little bit, but then they regress. FAP-P2 does not affect growth in the aged mice. However, if she now depletes FAP-P2 and treats with the inhibitors against the BRAF oncogene, she completely causes those tumors to regress and they stay gone for a really long time. So this is a very, I think, striking example of how targeting a molecule in an age-related manner can really overcome resistance to therapy and affect outcome in these mice and presumably in patients when we get there. All right, so thank goodness I'm at the point where I'm going to sum up, because I'm not sure I'll make it through the rest of the talk. But I hope that, I've convinced you that the aging microenvironment affects key aspects of tumor progression, and that it's going to be really important to consider the aging microenvironment as we design preclinical studies and treat patients as well. It affects everything from the matrix organization, angiogenesis, metabolism, and drives metastasis as well. These are all the amazing people in the lab who do the work. This is a group that moved with me from Philadelphia. These are all of our new additions. I always end every talk I give. And these are our wonderful collaborators across the street, across the world. Melanoma is a very global disease. Lots of people working on it all over the world. This is my favorite aging study. I always end talks with a picture of her, and she's surviving quarantine along with the rest of us. And then I'll just, for 1 second, if you'll indulge me, I will tell you how important it is for us to think about diversity in science. If you look at even just bachelor's degrees earned in STEM, less than 3% of Black women, 4% of Latino women, and 5% of Asian women even earn a bachelor's degree in STEM, let alone an advanced degree. If you're interested, my friend Anita and I just wrote this piece in Cancer Discovery. It's got a lot of great websites and resources. I strongly encourage you to give it a look. And with that, I'll stop, take any questions.
Great. Thanks so much, Ashley. That was really fantastic. Haida, are you on, or do you want me to read yours? You're the one person that gets an excuse because you're driving or in the car. I'll go ahead and read Haida's because okay. I don't know if she's in contact right now. So Haida asks I should mention Haida was the co-host of Ashi, and I forgot to mention that early on. Is the change in Wnt5a specific to changes in the H lung microenvironment, or does this also occur in other microenvironments such as the liver, et cetera?
Great question. So we've looked at fibroblasts in the skin, the lymph node, the lung, and the liver. The lung and the liver look a lot alike. The skin and the lymph node look a lot alike. So so the answer to your question is we're seeing some of the same changes in the liver, but it's the opposite in the lymph node, for example. So it really is organ site specific.
And I have a follow-up from that, from the kind of the opposite perspective, because you talked about dormancy, which I thought was really fascinating, how the aged fibroblasts promote sort of our anti-dormancy. Right. And that could be particularly relevant for breast cancer, because breast cancer has been shown, a woman can, think she's cured of breast cancer, and 20 years later it relapses, and it's often in the lung. And of course, that woman is now 20 years older. Right. So have you looked at this in the context of breast cancer, particularly related to dormancy?
Yeah, so we're looking actually with Julio Guiraguiso, who works on dormancy in breast cancer. So he's helping us look in melanoma. We're looking with him at breast cancer. Melanoma is the same, right? A lot of people get primaries removed when they're very young from their ankle. Usually it's a lower extremity like an ankle or a calf. And then 20, 30 years later, it comes back in their lung. I think one of the most striking dormancy experiments, as you may know, is the one where they have seen transplant patients, right? Where the primary melanoma was removed, that patient passed away of, I don't know, a car crash or whatever, donated their organs, and then the transplant recipient ends up developing melanoma that can be cytogenetically traced back to the original.
Yeah, no, definitely. And that could indicate that it was being kept in check by the immune system.
By the immune system, exactly.
Yep. Very cool. All right, we'll move to, so Christian, do you want to ask your own question, or do you want me to read it?
I can read it. It also gave me a chance to thank Ashley for a fantastic talk. It was just wonderful. So you kind of swung back to this a little bit, but I'd like more of your thoughts relating to what is the mechanism that is like differentially programming the fibroblast Wnt status in the lung versus the skin? Or what are your thoughts?
That's a great question. Where So I don't know whether it's because the fibroblasts and the lungs don't have positional stability. They're, turning over. So, and therefore secreting and or exposed to different environmental stresses than the skin is. We really don't have a good take on that part of it. So we've been looking down here and we don't have a good take of why that's happening up here.
But you're definitely thinking structure. That is your...
I think it is because it's so organ specific.
Yeah. Interesting.
And, and I don't know. Yeah, that's exactly right.
Great.
Sorry, that was a terrible answer to your question, but I just don't know.
It was a wonderful answer. Wonderful talk, wonderful answer.
Thank you.
So Harry, and if people don't mind, if you don't mind turning on your camera when you ask your question, I always think it adds a little bit of a personal touch. Harry, if you want to ask your question.
Sure. So regarding the increased uptake of fat, what would be the implication of it to the energetics? Do aged cells actively utilize fat as an energy resource, or does fat just simply accumulate?
That's a great question. So actually what we showed, and that's in the paper too, is that when they take up this fat, or at least when we have activated versus inactivated FAT-P2, they switch from a glycolytic to a more oxidative metabolism. And that oxidative metabolism allows them to sort of survive the BRAF inhibitors. It was striking because when we got rid of FAT-P2, whether using lipofumata or doxycycline, it acted as a mitochondrial poison almost. It looked like these cells just went straight into a mitochondrial crisis.
Great. Thanks. So Phil?
I'll click on the video. Great talk. Really inspiring work connecting the matrix, the vasculature, and the tumor and the microenvironment. It's really exciting. And so the question I have is related to that weakened lymph structure. And maybe you see a difference in blood vessels versus lymph vessels. And, it's kind of thinking of that input output as you get older and your ability to have better circulation, and how, if we're trying to get these immunotherapies to work, how that reduced efficiency perhaps in the ability of leukocytes to sort of traffic and activate antigens and process, and to get all these wonderful immunotherapies to work. Do you think that there's that it's just harder to traffic and drain out through a lymph if there's not a really good lymph drainage tube to a node?
Right. So that's exactly actually where this began, is because Brett, who was a surgeon in the lab, said, it's really interesting when I give technetium dye when they're doing sentinel lymph node imaging to a young patient versus an older patient. He's like, it's so easy to detect the node. So just detect the dye. I'm not even talking about tumor cells. In the young patient and the old patient, it's so hard for us to get a signal in the lymph node. And that's where the whole idea of, is it just leaking out along the way came from. So that's one part of your question. Yes, we think that, they're just leaking out. The structure is much weaker. In terms of immunotherapy, it's interesting because actually older patients do much better in response to anti-PD-1 and checkpoint inhibitors than younger patients. And part of it is because younger patients have more Tregs in their primary tumor. And so something that the lab is working on right now is looking at whether what is happening in the lymph node is educating the primary tumor. So in a young lymph node, if a tumor cell is going into the node, getting there and staying there, it's giving a signal to all of the immune cells in that lymph node, right? It's telling them, hey, there's something going on, there's a problem here. And then there's trafficking to the tumor site of signals. And so the question is, is that what's happening? Is there a response that is alerted and ignited in the young tumors because they see the lymph node? And that's why they need to regulate it with Tregs, and that's part of why they have more Tregs. But in the age, the tumor cells aren't really getting there, so they're not creating that initial response. But that in a weird way makes them more susceptible to the immune checkpoint therapies.
That's a great answer. Thank you so much.
You're welcome.
Great. And Christian, I see you have a follow-up question, which I'm actually really interested to get the answer on too.
Yes, this is a great question. So it's actually the part of aim 2 of our PM1 project. So it turns out that the ferroptosis is actually occurring in the myeloid-derived suppressor cells in the tumor microenvironment. And Dmitry Grbilovich, who was at Worcester, is now at AstraZeneca, was the co-leader on my project when he was in academia. He had shown that, myeloid-derived suppressor cells also have FAP-P2. They also take up more lipids. And then when you treat them with things like lipofumata, you can actually inhibit tumor growth. And part of the whole lipid uptake in the myeloid-derived suppressor cells was triggering this ferroptosis mechanism, right, when they treated them. We have not seen that in the melanoma cells yet, but we have also not investigated it as thoroughly as we should have.
Thank you.
If that makes sense.
Great.
And he had a really nice Nature paper on that if you want to take a look at that.
So I've got one other question, and that's it. Most times when you think about aging, everyone, of course, turns to inflammaging.
Yes.
And have you seen connections? I think when I've seen earlier talks that you did have some inflammatory cytokines being secreted by the fibroblast. But what is the crosstalk between that inflammation and the Wnt pathways?
Oh, that's a good question. And that's something I should know, and I don't really know. But, we are seeing for sure things, Natalie has shown beautifully things like CXCL12. We're seeing that upregulated in the age-long microenvironment. We think it's, arginase 1 is another one. So we do think that's part of why we're getting this MDSC response. We just had a paper where it's not related to aging, but we showed that the largest source of Wnt5a in the tumor microenvironment, much to our surprise. So you get Wnt5a from fibroblasts, you get it from macrophages, but one of the largest sources is the myeloid-derived suppressor cells. And they are just secreting tons of Wnt5a. We made a knockout mouse where we depleted Wnt5a from specifically the MDSCs. And when we do that, there's no Wnt5a in the tumor anymore. The irony of that is that they actually grow better because they don't have Wnt5a suppressing their growth. Even though they, have more immune infiltration, they're still growing better because the Wnt5a kind of overrides the immune effect, but they don't metastasize as effectively anymore.
Yeah,
so
but that's going to make it
definitely a lot of crosstalk.
Yeah, it's going to make it hard to target that pathway because
oh my
obviously.
God, it's terrible,
Yeah.
right? Because that's the thing. Like we always thought of Wnt5a was this dry, the nuances of how it's going to the lung and the plasticity. It makes it impossible to target because what if you had a few dormant cells and you target the primary tumor, you make it resistant to you make it sensitive to therapy, but now you've got all these lung mets growing out.
No, exactly. I think it shows you we've got to get smarter.
Yeah.
Use blunt instruments. That's good.
Yes, this tumor is way smarter than we are right now. That's for sure.
So does anyone else have any questions for Ashi? Speak now or forever hold your peace. We had a great turnout. I don't know if you caught the numbers down below, but it was close to 80 at some point, which is, I always worry people are going to get the Zoom exhaustion and stop tuning into seminars, but that has not happened yet. Or I think it was you and your title that brought them back. Anyway, Ashi, it was so fantastic. That was
Thank
wonderful.
Thank you so much.
And
thank you.
Hopefully the next time we'll actually have you here in person.
Yes, that would be lovely.
Yeah.
Take care. Thank you all.
Thanks everyone for joining in.
Everyone.
Bye-bye, Ashi. See you.
Take care. Bye.